Good afternoon. Thanks for attending my talk. I'd also like to thank the CannMed organizers for inviting me here today. The background image on my title slide is actually not related to the core of the talk, but I thought you might like to see it. This is a fossilized cannabis leaf dating to the Oligocene era, approximately 30 million years ago. The title of my talk is not classifying cannabis with genetics, but that's okay. We're going to get into that a little bit anyways. The title is Ethnobotanogenomics in Cannabis. And I'm going to focus on the genomic consequences of economics and law as forces of selection during cannabis domestication. Ethnobotanogenomics is a word that I made up. It's a portmanteau of ethnobotany, which is the study of plants and human interactions, and genomics, which is the study of genomes. So a key resource in genome research is, of course, a reference genome. And so I'll spend a little bit of time talking about the reference that I assembled and released last year. The project design included deep sequencing of 3 haplotypes and 2 components. First, the marijuana by hemp cross, and the genome assembly and physical mapping of CBDRX. The genetic map was based on the sequencing of a recombinant F2 population developed by George Weiblin's group at the University of Minnesota. First, 2 near isogenic lines were developed by inbreeding skunk number 1 and Carmen for several generations to make their genomes homozygous. These were crossed to produce a hybrid F1. An F1 female was hermaphrodized using colloidal silver and then self-pollinated. Seeds from the F1 grew into the recombinant F2 population, which we sequenced with whole genome low coverage Illumina. We also measured important agronomic traits in the F2 population, such as cannabinoid profiles and plant architecture. In this slide on the right, I show the genotypes, which are indicated in rows, of 96 of the F2s, each of which is indicated by a column in that square on the right. So this is a chromosome where you see blue, those regions of the genome inherited from the marijuana grandparent, and yellow is regions of the genome inherited from the hemp grandparent. Green is heterozygous regions of the genome, and transitions in color indicate where crossover events happen during meiosis in the F1. So each row, again, is a unique segregation pattern of genes in the F2 population, which you can correlate with the segregation of traits to determine the regions of the genome contributing to those traits. The core genome assembly was based on the sequencing of a highly inbred line from functional remedies called CBDRX. Todd Michael at the JCVI generated a total of 100x coverage of the CBDRX genome with Oxford Nanopore and Illumina reads. The Oxford Nanopore MinION is a USB device about the stick of gum. The MinION contains protein pores embedded in a glass plate. Electrodes measure the electrical resistance as DNA molecules pass through the pore, which is used to model the DNA sequence. The MinION generates the longest reads of any DNA sequencing technology, and long reads are essential for assembling repeat-rich plant genomes, but have a higher rate at the nucleotide level. So we assemble the long reads and the long contigs to get the genome structure right, then polish the contigs with highly accurate, high-coverage Illumina data to perfect the nucleotide sequence. We did further scaffolding with a high-C library from Phase Genomics and ended up with a little less than 2,000 contigs that form the basis of the physical map. So then I integrated the genetic and physical maps to produce chromosome-scale pseudomolecules corresponding to the 10 chromosomes of cannabis. The animation depicted in my slide shows each chromosome in white, with the gene's genetic position on the y-axis, shown in centimorgans, and the physical position shown in megabase pairs on the x-axis. I also show the recombination rate in red. It's very important because it tells us how tightly linked 2 positions in the genome are, which is very valuable for designing breeding experiments involving marker-assisted selection and for evolutionary inference, which most of my talk will be on. Note the extreme recombination suppression in the center of the chromosomes. About 90% of the genetic recombination takes place in about 10% of the genome. So the CBDRX genome was released late last year and has proven a valuable resource for the cannabis community. One of my favorite citations is this recent preprint that identified the sex chromosome as chromosome 1. In their main figure, they show the expression of sex-specific genes in red and blue bars. There are now several whole genomes available for cannabis, and we can use them to cross-validate each other. On the right is a figure that Kevin McKernan presented yesterday in his talk. It's a syntenic plot of the Jamaican lion assembly against the CBDRX assembly. These diagonal lines indicate a high level of agreement and validate the structure of both. So now I'm going to switch gears a little bit and put on my evolutionary biologist hat. I'd like to review some key concepts that will be helpful for the rest of my talk. At the center of the slide is phenotype, the composite of an organism's traits, and what I'm guessing most of you are most interested in. The plant's phenotype is controlled in large part by its genes. Definition of evolution that I'm using today is change in allele frequency over time. And an organism's interaction with its environment is called the niche, the ecological niche. These interactions are influence a reproductive success to a great degree and thus drive evolution at the population level. An ecological niche includes its habitat, such as precipitation, temperature, day length, soil nutrients, as well as interactions with pollinators and predators. Now, the ecological niche of domesticated organisms also includes its interaction with human culture, how it's used, the demand for its product, how the product is regulated. Here's a picture of some wild cannabis growing in its native habitat in the Eurasian steppe. This plant is well adapted to a weedy life history on this riverbank, but it doesn't look like a particularly valuable crop. It's short and branching and makes small flowers. Over the past few millennia, humans have selected for composite traits very different from this, and in the process domesticated two very different crops. Here's a picture of some fiber hemp. It's very tall, doesn't branch much, and can be mechanically harvested with ease. Most of the biomass is allocated to the stems, which the fibers are harvested. Cannabinoids are typically undesirable in fiber hemp as they gum up the processing machines. Most hemp is not very intoxicating. So contrast this with marijuana, pictured here. In general, marijuana is relatively short, branches more, allocates a large proportion of biomass to its flowers. The majority of our precious cannabinoids are secreted into glandular trichomes. Here's a principal component analysis, or PCA, of genetic data from about 300 individuals from 4 different studies. A PCA is a statistical technique for compressing millions of genetic variants down to a few dimensions, or principal components, that explain the most variance in the data. Each individual is represented by a single point in the plot and is colored according to its functional class. The plot shows the top 2 principal components. You can conceptualize the y-axis as how far along the individual is in the domestication process. The x-axis corresponds to the primary principal component and clearly separates marijuana from hemp. As humans selected for different phenotypes to process into different products, the populations evolved genetically. The plot below is a more granular view of each individual's ancestry. Each column in the lower plot is a stacked bar plot showing the percent ancestry in its genome from each of the 3 functional classes. Here's another way to look at population differentiation. This is a population branch statistic, which is based on how similar allele frequencies in populations are. The longer the branch, the more differentiated the populations are from each other. Notice the small inset tree near the histogram. This shows the genome-wide average of the population statistic. We know from the previous slide that marijuana and hemp are highly differentiated, and that is reflected in the branch lengths. The large tree, however, is drawn in the same scale as the small tree, which shows the population branch statistic at the synthase locus. It's an extreme outlier. When you look back at the histogram that shows the distribution of branch lengths in the marijuana line, I've indicated the 99.95th percentile in the dotted line, and shown with an arrow where the synthase locus lies in that distribution. What this tells me is that there's been strong divergent selection for very different phenotypes or alleles producing those phenotypes at the synthase locus. If we look at the landrace or the natural populations, we actually have a negative branch length suggesting that the locus evolves neutrally in the wild. It's not under strong natural selection. So what about high-CBD cannabis? High-CBD cannabis plants typically have mostly marijuana-like phenotypes. They're shorter, more branching, they have very large flowers, and are high in total cannabinoids. However, they are low in THC and are non-intoxicating. They have a mosaic phenotype of both marijuana-like and hemp-like traits. How did we get here? This animation shows changes in cannabis law in the United States through time. In the past 5 years, laws have been passed progressively legalizing CBD. As we all know, last year's Farm Bill legalized hemp and CBD. The law classifies hemp as a cannabis plant that produces less than 0.3% THC. Here's a bar plot showing a steady increase in CBD sales over time. My core argument here is that these two changes, legalization of CBD coupled with a steady increase in sales, opened up an unprecedented ecological niche. That is, a new way for cannabis to interact with human culture. This is a genome plot for the 10 CBDRX chromosomes. Remember how I mentioned that the genetic map could be used to identify regions of the genome responsible for functional traits? On this plot, I show the loci responsible for traits pertinent to producing the marijuana and hemp phenotypes. Loci responsible for plant architecture are indicated by green and gold stars. Total cannabinoids by black stars, and the synthase locus with the red star. I also colored the chromosomes according to their ancestry. Regions of the genome inherited from marijuana ancestors are colored in blue. Regions inherited from hemp ancestors are colored in yellow. We can see that the CBDRX genome is about 90% marijuana derived and about 10% hemp derived. So just as high CBD plants have mosaic phenotypes, they also have mosaic genomes. Mosaic patterns are the same. Here's a graphical summary of the concept I just discussed. Marijuana and hemp have long been selected for very different phenotypes and have diverged genetically, as indicated by the 3 colors. The law, however, is only concerned with percent THC, and the legal classification of plants is indicated by the style of shading. The breeding history of high CBD cannabis varieties involved the integration of hemp-like synthase loci into a predominantly marijuana-like genomic background. This is a schematic of my mental model of population dynamics in cannabis. The right-hand side illustrates the domestication process, which we have covered. And I'm also interested in the global patterns of biogeography and dispersal. I'll conclude my talk today with some preliminary analyses of Old World biogeography. I scraped some genetic data provided by the now defunct Open Cannabis Project and clustered approximately 100 individuals based on shared ancestry. In this matrix, each row or column is a single individual. Cells are colored according to the proportion of shared ancestry, with yellow indicating low level of shared ancestry, moderate shared ancestry in red, and high in pink or blue. In this plot, I intentionally omitted elite hemp and marijuana cultivars in order to highlight the differences between land races. The samples loosely cluster into 18 groups and show a pattern of isolation by distance. The most differentiated clusters are those of Central Africa and Central Europe. Notice the tree at the top of the plot. I overlaid this tree from the previous slide onto a geographic map to get this figure. The red arrows show hypothesized dispersal routes by which cannabis expanded its range. You can also map the Old World physical biogeography on a physical map of the genome. In this animation, I colored genomic segments according to the predicted ancestry, found the elite hemp cultivar to be descended primarily from the Slavic lands of Central and Eastern Europe, while the elite marijuana cultivar is descended mostly from South Asia and the Middle East. That's it. Thanks for attending my talk. I'm at Harvard University. Most of this was done as a consultant for Sunrise Genetics. The genetic mapping population was developed at the University of Minnesota by George Weeblin. Todd Michael at the JCVI produced most of the core sequencing data. Functional Remedies of Colorado provided some funding, and the CBDRX. And of course, none of this would have been possible without all these amazing sequencing technologies. So that's it. Well, I'll take questions. Hey.
I was curious, looking at the range of phenotypes you were covering there in the genetics, do you have any sense of whether you feel like there's limits on kind of extreme phenotypes that we can get out of the plants? Is that something
No.
predictable?
I don't think that there's a limit on it. If you look at like maize yields over the past century, there's been a linear relationship between yield and time. So I think if we keep pushing it, the plants will respond.
Thank you.
First of all, fascinating talk.
Thanks.
I'm curious if you could go sort of back in time, like what were sort of the original selective pressures that you've identified from that wild type that you showed a picture of that was essentially nothing that anybody would resemble, nothing that anybody would consider to be
So I mean, Cannabis sativa is, I consider it a single species with a lot of diversity. The native range is probably like the Eurasian steppe, and so depending on where the population was located, that would dictate kind of what it was adapted to. So we see extreme variation in day length and precipitation, and actually, all of this wild germplasm potentially could be a great source for breeding if we can collect the seeds.
Thank you.
You're welcome. Okay.